Hi, I'm Pushpangna Singh. I am a PMRA fellow from IIT Kanpur in the May 2020 cycle, and the lecture that I'm going to focus on today is on HIV or human immunodeficiency virus. In the late 1970s and 1980s, previously healthy patients with symptoms of immunologic dysfunction sought advice and treatment from physicians in the United States and Europe. This new and unusual syndrome was characterized by generalized lymphadenopathy, opportunistic infections like that of cytomegalovirus, meningitis, etc., and a variety of unusual cancers such as non-Hodgkin's lymphoma and Kaposi sarcoma. A common accompanying laboratory in affected individuals was marked depletion of the CD4 plus T lymphocyte subset in the peripheral blood. The disease was first brought to the attention of the general medical community in June 1981 when the Center for Disease Control and Prevention described California men with severe immunodeficiency in the Morbidity and Mortality Weekly Report. This notification was followed by several reports describing male homosexuals and intravenous drug users with impaired immune systems characterized by T lymphocytes that responded poorly to antigen and other immunogen stimulation in functional assays. Within several months, it became clear that a similar immunodeficiency syndrome was also affecting other groups, including hemophiliacs, blood transfusion recipients, recent Haitian immigrants, and most significantly, sexual partners or children of members of the other various risk groups. The emerging epidemiological pattern suggested that the new disease was transmitted by a novel pathogen in contaminated blood or following sexual intercourse with an affected individual. Between late 1981 and early 1983, a variety of microorganisms were proposed as possible causes of acquired immunodeficiency syndrome or AIDS, as the disease was soon called. In the spring of 1983, Montagnier and colleagues at the Pasteur Institute reported the isolation of an agent from the lymph nodes of an asymptomatic individual who presented with generalized lymphadenopathy of unknown origin. During its replication in cultured cells, the lymphadenopathy-associated virus, or LAV as it was named, released high titers of progeny virions that contain magnesium-dependent reverse transcriptase or RT activity and exhibited electron microscopic features typical of retroviruses. However, unlike the commonly studied retroviruses such as the avian leukosis viruses ALVs and the murine and feline leukemia viruses MLVs and FELVs, LAV was highly cytopathic in human peripheral blood mononuclear cells or PBMCs, specifically killing CD4 plus T lymphocytes in cell cultures. Gallo and colleagues at the National Institute of Health or NIH subsequently reported the isolation of a retrovirus from an AIDS patient, which they named human T leukemia virus type 3 or HTLV3 to distinguish it from non-cytopathic HTLV and obtain the first serological evidence linking exposure of ALV like retroviruses and immunodeficient individuals from the various groups at risk. The new retrovirus associated with AIDS in the United States, Europe, and Central Africa and exhibiting morphological and genetic characteristics typical of the lentivirus genus was named human immunodeficiency virus or HIV and subsequently HIV-1. In 1986, a related but immunologically distinct human retrovirus, now called HIV-2, was recovered from individuals residing in several West African countries such as Senegal, the Ivory Coast, and Guinea-Bissau. Prior to the discovery and use of highly active antiretroviral therapy or HAART, The risk of death for HIV-1 infected person was 40-fold higher than the mortality in the general population, whereas HIV-2 infected individuals had a 2 to 5-fold higher risk. So what exactly is HIV? HIV, as I have mentioned earlier, is human immunodeficiency virus. Human is because this particular virus can infect only human beings. Immunodeficiency is because HIV weakens your immune system by destroying important cells that fight disease and infection. A deficient immune system can protect us. And a virus can only reproduce itself by taking over a cell in the body of its host. The disease that HIV causes is AIDS, or acquired immunodeficiency syndrome. Acquired is because AIDS is not something that you inherit from your parents like eye color. You acquire AIDS. Immuno is your body's immune system includes all the organs and cells that work to fight off infection or disease. So it's immunodeficiency. You get AIDS when your immune system is deficient or isn't working the way that it normally should. And syndrome is a collection of symptoms and signs of disease. AIDS is a syndrome rather than a single disease. It is a complex illness with a wide range of symptoms. So this is the basic structure of HIV virion. It has this envelope which has the HIV capsid, HIV RNA and HIV enzymes, and it also has glycoproteins or spikes which are responsible for the first line of interaction with the host cell. So HIV capsid is the code that contains the HIV RNA which has the reverse transcriptase, and HIV envelope is the outer surface of HIV which protects all its inner contents. And there are various other HIV enzymes that carry out the important steps in the HIV life cycle. Glycoproteins are the spike that are embedded in the HIV envelope, and HIV RNA is HIV's genetic material. Now, one of the first features of Pasteur Institute that the Pasteur Institute group noted about HIV was its particle-associated RT activity, a property that placed the new agent in the Retroviridae family. Now, this feature was consistent with the EM analysis of particles released from infected cell cultures, which revealed 100 to 120 nanometer envelope virions, similar in size and morphology to previously studied retroviruses. The mature HIV-1 particles contained a cone-shaped cylindrical core reminiscent of that previously described for other viruses. Now these two are electron micrographs that show a single human immunodeficiency virus 1 particle in the process of budding from the infected culture peripheral blood mononuclear cells or PBMCs from humans, and several mature virions containing the characteristic conical or bullet shaped cone as you can see here. And this EM snapshot on the right shows typical ballooning syncytia induced by primate lentiviruses as visualized by inverted light microscopy at the time of peak virus production. So this is the basic organization of the HIV genome, and it is composed of 9 genes which code for 15 proteins, and these genes code for these respective proteins which have been shown here in this diagram. And for comparison, we can say that the E. coli bacterium contains around 4,377 genes, and the human genome encodes around 21,000 genes. So HIV is that small. Now the genes of Molyneux-Murine leukemia virus or MLV, human T-cell leukemia virus or HIV, and HIV-2 are depicted here as they are arranged in their respective proviral DNA. The sizes of different proviral DNA are shown in proportion to the 9.7 kb HIV virus. Nucleotide sequencing of several of the original HIV-1 isolates revealed an unexpected result. In contrast to the well-characterized and intensively studied prototypical retroviruses such as ASLV and MLV, whose genomes contain only three genes, that is GAG, POL, and envelope, encoding the structural proteins and enzymes required for productive infection, the HIV-1 genome includes several additional and overlapping open reading frames or ORFs of unknown function. Not only were HIV-1 and HIV-2 shown to contain multiple additional ORFs, but also their genome organizations appear to be very similar. Further, analysis revealed that HIV-1 contained the distinguishing VPU gene and HIV-2 carried a signature VPX gene. Like all replication competent retroviruses, the three primary HIV-1 translation products, all encoding structural proteins or enzymes, are initially synthesized as polyprotein precursors, which are subsequently processed by viral or cellular proteases into mature particle-associated proteins. The 55 kilodalton GAG precursor, or the P55 GAG, is cleaved into the matrix, capsid, nucleocapsid, P6, and two spacer peptides sp1 and sp2 during or after the release of progeny virions. Autocatalysis of the 160 kilodalton GAG polyprotein gives rise to the protease, the heterodimeric RT, and integrase protein. Whereas the proteolytic digestion by a cellular enzyme converts the glycosylated 160 kilodalton envelope precursor GP160 into GP120 surface and GP41 transmembrane cleavage products. The remaining 6 HIV-1 encoded proteins VIF, VPR, REV, VPU, and NEF are the primary translation products of spliced messenger mRNAs. HIV-1 and HIV-2 have incorporated multiple sequence elements into their genomic RNAs that direct the balance and coordinated production of progeny virions. Many of these cis-acting RNA elements, as we can see here, are present in other retroviral genomes, but a few are unique to the primate lentiviruses. The 5' around 330 nucleotide untranslated region of HIV-1 genomic RNA is highly structured and contains multiple elements that mediate transcriptional elongation of viral RNA transcripts, splicing, genomic RNA dimerization, packaging of full-length viral RNA, and reverse transcription. A schematic model of this 5' leader sequence deduced from genetic, functional, structural, and nuclease accessibility mapping elements is shown here. Now, three regions, which are shown here in A, B, and C, encompassing the primer binding site or PBS stem, participate in the placement and stabilization of transfer tRNA lyse 3 primer, which is incorporated into HIV-1 particles and is required for the initiation of reverse transcription. These regions include the PBS itself, which can base pair with the 3' terminal 18 nucleotide long of tRNA lyse 3, the primer activation signal, which is PAS, which is shown here, which interacts with the thymidine pseudouridine cytidine TYC arm of tRNA to trigger the reverse transcription reaction, and the A loop, which is complementary to the anticodon loop of tRNA lysine 3. The unwinding of both the tRNA primer and the PBS stem within the 5' untranslated region, or the UTR, of the HIV-1 genome, as well as the incorporation of the primer into nascent virions, is facilitated by the NC domain of the GAG polyprotein, which plays the role of a molecular chaperone in the process. The packaging of HIV-1 genomic RNA into progeny virus particles is also directed by the NC domain of the GAG polyprotein and is primarily dependent on its interaction with the 4 RNA stem loops SL1, SL2, SL3 and SL4, constituting the core encapsulation signal Y. It is currently thought that a fully functional HIV 5 packaging signal may include the entire 5' UTR plus the first 300 nucleotides of the GAG gene. In virions, the HIV-1 genome is present as an RNA dimer, non-covalently linked at the 5' end of each molecule. Particle-associated dimeric RNA provides an alternative template for reverse transcription should the enzyme encounter a nick or break during the polymerization reaction. The dimerization initiation sequence or DIS located at the crown of SL1 contains a complementary exposed and palindromic sequence. Most of the non-B/D HIV-1 subtypes, including the highly prevalent subtype C viruses, have a GTG-CAC for their DIS, whereas clade B and D subtypes have a GC-GC-GC in their DIS. Dimerization is initiated via kissing loop mechanism involving base pairing between the palindromic sequences present on paired HIV-1 genomes. Other regions of the SL1 participate in stabilizing the RNA dimer into a more extended duplex structure, a process also believed to be chaperoned by NC. The 5 packaging signal domain also contains the major splice domain or the MSD near the crown of SL2 and the AUG GAG start codon at the base of SL1. It has been proposed that the HIV-1 leader can switch from multiple hairpin structure as shown here in A to an alternate rod-like conformation, and that each conformer specifically promotes either packaging or translation of the viral RNA. In this model, an individual stem loops are formed as intermediates during RNA synthesis, but the entire HIV-1 leader is eventually converted to the thermodynamically more stable rod structure containing an exposed AUG, GAG start codon and an occluded DIS motif. The rod conformer containing a masked DIS may promote the translation of HIV-1 mRNAs into viral encoded proteins. A subsequent switch to the alternative conformation mediated by NC would expose the DIS and phi elements, favoring dimerization, packaging and ultimately reverse transcription of the viral genomic RNA. Evidence for the existence of alternative RNA conformers in virions or infected cells and the hypothesized conformational switch is still forthcoming. In HIV-1, the AAU-AAA binding site for cleavage and polyadenylation factor or CPSF and the GU/U region binding site for the 3' terminal cleavage stimulation factor CSTF are both situated within the repeat or the R regions at each end of the viral genomic RNA. An active 5' poly A site would obviously compromise the generation of long RNA transcripts by mediating premature cleavage and polyadenylation. A polyadenylation enhancer situated upstream of the 3' poly A signals stabilizes the binding of CPSF to AAU-AAA and promotes poly A addition, whereas the 5' poly A stem or the closely positioned MSD has been reported to suppress polyadenylation activity. Lentiviruses are unique among retroviruses in having a novel polypurine tract or PPT in addition to the canonical U3 proximal PPT, both of which are used for synthesis of plus strand viral DNA. The viral genomic RNA also contains a heptameric 7U, a slippery sequence within the GAG gene, where ribosomal frame shifting occurs. This sequence functions in conjunction with a downstream 8 nucleotide spacer and hairpin to mediate -1 translational frame shifting. The viral RNA also folds into two additional complex structures, the transactivation response region TAR and Rev response element RRE, which are involved in RNA synthesis and RNA nuclear to cytoplasmic transport respectively. Multiple inhibitory sequences INS associated with instability or nuclear retention of HIV transcripts are scattered throughout genes encoding the GAG, POL and envelope genes. Finally, although not strictly considered a cis-acting element, HIV-1 genomic RNA contains a significantly higher adenosine deoxyribonucleic acid content, approximately 39%, than the mammalian DNA. This bias contributes to an unusual codon usage. The secondary structure of the entire HIV-1 genome has recently been determined using a technique called selective 2' hydroxyl acylation, analyzed by primer extension or SHAPE, which we can call in short. SHAPE is based on the chemical modification of unpaired nucleotides in RNA molecules and the resultant termination of reverse transcription at the altered site. Regions of RNA with significant secondary structures are not readily modified, whereas looped out RNA segments exhibit high shape reactivity. This allows the determination of the folded state of RNA using a computer algorithm. Some of the tightly base paired RNA regions identified by SHAPE correspond to previously described TAR structures at the 5' and 3' termini of the HIV-1 genome. The SP1 encapsulation site in the 5' UTR and the RRE. Interestingly, structured RNA segments were also present in protein coding regions located between individual GAG, POL and envelope subdomains, which give rise to mature protein cleavage products. In addition, base paired regions of HIV-1 RNA are located adjacent to sequences encoding protein domains that undergo co-translation folding or unfolding. Ribosomal pausing induced at these structured RNA segments may retard translation to allow sufficient time for a nascent protein to attain its final conformation. Non-structured RNA regions encoding the GP120 variable loops were enclosed by stable RNA duplices, yet another example of correlated RNA and protein structures. So this shows the HIV life cycle. So the main 7 stages of HIV life cycle are: first is binding or attachment, second is fusion, third is reverse transcription, fourth is integration, fifth is replication, sixth is assembly, and seventh is budding. So this life cycle begins with the binding of the virus particles to CD4 molecules on the surface of susceptible cells. In the context of tissue culture systems, susceptible cells include activated CD4 plus T lymphocytes present in PBMC collected from uninfected individuals, T cell leukemia cell lines, and MDMs. In vivo, activation of CD4 plus T cells is not required for HIV-1 replication. Although binding of virions to CD4 plus cells is essential for HIV infectivity, their subsequent interaction with a co-receptor, now known to be members of the 7 membrane spanning CC or CXC families of chemokine receptors, is required for membrane fusion and entry. Multiple chemokine receptors exhibit activity in fusion or entry assays with HIV-1 GP120. However, the two most physiologically important co-receptors are CXCR4 and CCR5. Unlike some other envelope viruses that enter cells by receptor-mediated endocytosis, HIV-1 and many other retroviruses fuse directly with the plasma membrane. Once inside the cell, HIV-1 encounters a cortical actin barrier, which can be remodeled by the virally encoded NEF protein. Partially uncoated virions migrate from cortical actin to the microtubule network, where reverse transcription is initiated and movement to the nucleus begins. As with other retro elements, the partially double-stranded DNA reverse transcription product is transported through the cytoplasm and to the nucleus as a component of the PIC, which contains a subset of the GAG and POL proteins. As noted earlier, the lentiviruses are unique among retroviruses in generating PICs that are actively transported by the nuclear import machinery into the interphase nucleus of non-dividing cells arrested in the G1 phase of the cell cycle. After the import of the PIC into the nucleus, full-length linear copies of the reverse transcript are integrated into the chromosomal DNA of the infected cell, a step that is required for efficient viral RNA synthesis and infectious particle production. In activated T lymphocytes, integrated copies of HIV DNA serve as templates for RNA polymerase II or Pol II directed viral RNA synthesis. The coordinated interaction of the HIV encoded TAT protein and the cellular NF kappa beta and sp1 transcriptional transactivating proteins with the RNA Pol II transcriptional apparatus ensures the production of high levels of viral RNA. Unspliced or partially spliced HIV transcripts are exported from the nucleus to the cytoplasm by a unique transport mechanism mediated by the virus-encoded Ref protein, which shuffles between these compartments. The subsequent translation of the GP160 envelope precursor occurs in the endoplasmic reticulum, whereas the GAG and GAG-POL polyproteins are synthesized on free cytoplasmic ribosomes, and each is independently transported to the plasma membrane. The GAG and GAG-POL polyproteins, in association with dimers of genomic RNA, condense at the plasma membrane to form an electron-dense bud that gives rise to a spherical immature particle containing the mature TM and SU envelope glycoproteins. Proteolytic processing of the GAG and POL proteins by HIV-PR during or immediately after particle release generates the cone-shaped core characteristic of mature HIV virions. Thus, HIV-1 uses the same replicative strategy as the so-called simple retroviruses, which encode only the GAG, POL, and envelope proteins. However, during its evolution, HIV-1 has acquired 6 additional genes to carry out functions that are either A. performed by cellular proteins already present in the cells infected by the simple retroviruses, or B. uniquely required for virus replication, transmission, and survival in hematopoietic cells targeted by the primate lentiviruses. Some of the HIV-1 accessory proteins such as VIF, VPR, VPU, and NEF are not required for replication in certain human T cell lines. Although virus infectivity may be affected up to several thousand folds depending on the accessory gene and the type of infected cell. In view of their conservation, however, these additional genes must be required for successful HIV-1 infectivity in vivo. HIV-1 encodes only 15 proteins and, like all other viruses, must utilize a large number of cellular proteins for successful replication. Over the years, several techniques such as the immunoprecipitation or mass spectrometry and East2 hybrid screening have been used to identify such host cell factors. The recent development of genome-wide small interfering RNA, siRNA, or short hairpin shRNA screening has provided genetic approaches to identify potential human proteins required for HIV-1 replication based on loss of function readouts. In these experiments, siRNAs are transfected into target cells, whereas shRNAs are delivered using lentiviral vectors and subsequently processed into siRNA intracellularly. Of the 842 genes identified as being required for HIV-1 replication in 3 siRNA screens, 34 were also reported in at least 2 screens and 3 were detected in all the 3 screens. The overlap between 3 siRNAs and single shRNA screen range from 0 to 3 genes in pairwise comparisons. 11 of the 34 genes found in 2 siRNA screens had also been previously reported to encode HIV-1 interacting proteins, and 29 of the 34 genes are expressed in cells susceptible to HIV-1, which are CD4 and chemokine co-receptor positive. The overlap between individual screens is unexpectedly small, yet is statistically significant. This result is not surprising given that different challenge viruses, cell lines, siRNA libraries, periods of gene silencing prior to infection, and readouts of infection were used in the 4 studies. Nonetheless, all 3 siRNA screens identified factors associated with transport through the nuclear pores, trafficking between cellular compartments, DNA repair, RNA binding, and interaction with the ubiquitin proteasomal degradation machinery. 2 of the siRNA screens identified previously reported cellular proteins known to be critical for HIV-1 infectivity, including CD4, CXCR4, REL-A, elongin B, and cyclin T1. Cyclophilin A, SP1, and lens epithelium-derived growth factor LEDGF are among the known HIV-1 cofactors that were not identified in any of the screens. The primary function of viral envelope glycoproteins is to promote a membrane fusion reaction between viral and target cell membranes. This membrane fusion event enables the viral core to gain entry into the host cell cytoplasm. The observation that CD4 induces conformational changes in GP120 that enhance interaction with co-receptors suggests that events leading up to membrane fusion occur sequentially. GP120 first interacts with CD4. A ternary CD4 co-receptor GP120 complex then forms, and initially conformational changes take place in the GP41 that trigger membrane fusion. Although fusion between the viral and cellular membrane is widely believed to occur directly at the plasma membrane after CD4 and co-receptor engagement, it has been reported that fusion can also occur in low pH endosomal compartment. Mutational analysis demonstrated that a hydrophobic region at the end terminus of GP41, the so-called fusion peptide, which is shown here, plays a central role in membrane fusion mediated by HIV-1, HIV-2, and SIV. C-terminal to the GP41 fusion peptide are 2 amphipathic heptad repeat HR domains that are also required for membrane fusion. Mutations in the N-terminal HR1 impair infectivity and membrane fusion activity, and peptides derived from HR1 and HR2 exhibit potent antiviral activity in culture. Structures for the ectodomain of HIV-1 and SIV GP41, including the HRs, were determined by X-ray crystallography and nuclear magnetic resonance NMR spectroscopy. These studies obtained fundamentally similar trimeric structures in which HR1 and HR2 pack in an anti-polar fashion to generate a 6-helix bundle. The HR1 form a coiled coil in the center of the bundle, with the HR2s packing into the hydrophobic grooves on the outside. The GP41 ectodomain structure resembles that of the fusion competent low pH induced form of influenza HA2, making it likely that HIV or SIV enveloped glycoproteins trigger membrane fusion by the same spring-loaded mechanism proposed for the influenza virus. Peptides corresponding to the HRs inhibit fusion and virus activity by interacting with their complementary binding partner on the GP41, thus preventing GP41 itself from forming a 6-helix bundle. Binding studies using native HIV-1 envelope indicate that GP41 interacts with HR2 peptide only after CD4 binding, suggesting that HR1 and HR2 undergo conformational changes after CD4 binding and that these rearrangements are required for membrane fusion. The cytoplasmic domain of GP41 also plays a role in modulating the fusion activity of envelope, in part by regulating the levels of cell surface expression of the GP120 GP41 complex and by influencing the conformation of GP120 and ectodomain of GP41. Whereas GP41 interacts directly with the lipid bilayer to catalyze the membrane fusion reaction, a variety of domains within GP120 are involved in activating envelope fusogenicity. The V3 loop of HIV-1 GP120, which elicits isolate specific neutralizing antibodies, is an essential player in membrane fusion. Mutations throughout the V3 loop of HIV-1 and the analogous region of HIV-2 block syncytium formation and virus infectivity. The importance of V3 in membrane fusion appears to be in part a consequence of its role in GP120 co-receptor interactions. The structure of the V3 loop in the context of the CD4 bound GP120 core reveals that V3 protrudes out from the core toward the target cell membrane and the co-receptor. It has become apparent that the lipid composition of the target cell plasma membrane plays an important role in membrane fusion. Of particular interest is the concept that membrane fusion may take place in plasma membrane microdomains known as lipid rafts. These microdomains, which are enriched in glycosphingolipids and cholesterol, appear to be involved in the entry of a number of envelope viruses, including HIV-1. Depletion of cholesterol from either the virus particle or the target cell membrane disrupts fusion, as does depletion of specific glycosphingolipids. The cholesterol binding compound amphotericin B methylester or AME is also a very potent inhibitor of HIV-1 infectivity. Interestingly, HIV-1 evades AME by acquiring mutations in the cytoplasmic tail of GP1 that cause the tail to be cleaved by PR, thereby allowing infection to proceed even in the presence of the inhibitor. In addition to promoting cell-free HIV-1 infection, lipid rafts may play an important role in HIV-1 transmission between cells and in virus uptake into DCs. By definition, retroviruses possess the ability to convert their single-stranded RNA genomes into double-stranded DNA during the early stage of infection process. The enzyme that catalyzes this reaction is RT, or reverse transcriptase, in conjunction with its associated ribonuclease H or RNase H activity. Retroviral RTs have two enzymatic centers: a DNA polymerase that can copy either RNA templates for minor strand DNA synthesis or DNA templates for second or plus strand DNA synthesis, and B, RNase H for the degradation of the tRNA primer and genomic RNA present in DNA RNA hybrid intermediates. RT was the first viral protein targeted by antiretroviral therapy, and RT inhibitors remains central to the treatment of HIV infected patients. In addition to providing an essential function in the virus life cycle, the enzymatic activity of RT is routinely used in the laboratory to quantitatively monitor levels of progeny virions present in the supernatant of infected cultures and to elucidate the molecular details of virus replication. Also, the detection of viral DNA post-infection by real-time polymerase chain reaction or PCR provides one of the most reliable methods to date for detecting entry steps in the virus replication cycle. Once inside the nucleus, some double-stranded viral DNA is ligated to generate circular forms. Although these circular DNA products do not integrate, their formation is often used to monitor nuclear import. The mature HIV-1 RT holoenzyme is a heterodimer of 66 and 51 subunits. The 51 kilodalton subunit or P51 is derived from the 66 kilodalton or P66 subunit or some larger precursor by proteolytic removal of the C-terminal 15 kilodalton fragment of P66 by PR. The structure of HIV-1 RT has been determined by X-ray crystallography in a number of studies. The enzyme has been crystallized in several contexts, unliganded, bound to a short DNA duplet, and the Fab portion of an anti-RT antibody, covalently linked to a complex of primer template and deoxyribonucleotide triphosphate DNTP, and bound to an RNA DNA duplet. A number of structures are also available for the RT bound to non-nucleoside RT inhibitors. The crystal structure of HIV-1 RT reveals that the P66 and P51 subunits are folded into similar subdomains, but these subdomains are arranged quite differently in P66 and P51. The P66 subunit can be visualized as a right hand with the polymerase active site within the palm and the deep template binding clefts formed by the palm, fingers, and thumb subdomains. The polymerase domain is linked to RNase H by the connection subdomain, which is this. The active site located in the palm contains three critical ASP residues, ASP110, ASP185 and ASP186 in close proximity and two coordinated Mg2 plus ions. Mutation of these ASP residues abolishes the polymerase activity of RT. The orientation of the three active site ASP residues is similar to that observed in other DNA polymerases. For example, the Klenau fragment of Escherichia coli DNA pol1. The P51 subunit appears to play a structural role and does not form a polymerizing cleft. ASP110, ASP185 and ASP186 of P51 are buried within the subunit. Approximately 18 base pair of the primer template duplet lie in the nucleic acid binding cleft, stretching from the polymerase to the RNase H active site. In the RT primer template DNTP structure, the presence of a dinucleotide DDNTP at the 3' end of the primer allows visualization of the catalytic complex block just prior to attack on the incoming DNTP. Comparison with the previously obtained structures suggests a model whereby after the addition of the incoming DNTP to the growing chain, the fingers adopt a more open configuration, thereby releasing the pyrophosphate and enabling RT to bind to the next DNTP. HIV-1 RNase H, whose structure has been determined by X-ray crystallography, displays a global folding similar to that of E. coli RNase H. HIV genomic information is in the form of positive RNA, but HIV replication involves an obligatory conversion of this RNA into double-stranded DNA that is incorporated into the infected host cell genome. HIV thus encodes for a specific enzyme, reverse transcriptase or RT, as we have discussed in the previous slide, to carry out this process. Reverse transcription initiates from an RNA primer provided by a specific cellular tRNA or tRNA lyase 3 incorporated during virion assembly. The 18 3' terminal nucleotides of this tRNA are annealed to a complementary sequence near the 5' end of the HIV genomic RNA termed primer binding sequence or PBS. RT-catalyzed RNA dependent DNA synthesis then proceeds until RT reaches the 5' end of the RNA genome, providing a strand of HIV minus DNA complementary to the U5 and R termini repeats of the HIV genomic RNA. These newly synthesized sequences are essential for hybridization to the 3' end of the HIV genomic RNA template to enable completion of full-length negative DNA synthesis. However, the negative sense DNA sequences are in the form of an RNA-DNA hybrid duplet. The RNA strand of this duplet must be removed to allow hybridization of the newly synthesized viral DNA with the terminal repeat region of the 3' end of the viral RNA. The RNase H activity of RT removes this RNA strand, enabling strand transfer and continuation of reverse transcription. If the RNA strand is not removed, reverse transcription and HIV replication stop. After the first strand transfer, RT-DNA polymerase activity continues negative DNA synthesis and RT-associated RNase H degrades the template RNA. During this process, a purine-rich sequence of HIV genomic RNA, the polypurine tract or PPT is generated. The PPT in duplet with complementary DNA is somewhat refractory to RNase H catalyzed degradation and serves as a primer for synthesis of HIV plus DNA strand. RT-RNase H removes the PPT RNA component after priming of positive DNA synthesis. Following a sufficient elongation, the PPT RNA component is degraded again by RNase H. Viral plus DNA synthesis continues, including that part of the tRNA initiation primer still associated with the negative DNA. RT-RNase H activity then acts to remove the tRNA component still associated with the nascent viral DNA. RT-RNase H activity is thus essential at several stages of HIV replication. A distinguishing feature of retrovirus replication is the integration or insertion of DNA copy to the viral genome into the host cell chromosome after reverse transcription. The integrated viral DNA or the provirus serves as a template for synthesis of viral RNAs and is maintained as part of the host cell genome for the lifetime of the infected cell. Retroviral mutants deficient in the ability to integrate generally fail to establish a productive infection. The integration of viral DNA is catalyzed by IN or integrase, a 32 kilodalton protein generated by PR-mediated cleavage of the C-terminal portion of the HIV GAC-Pol polyprotein. The steps in the integration process were originally elucidated in studies using MLV, but these findings apply to HIV as well. In all retroviral systems, integration proceeds in the same series of steps. So the first step is the 3' processing. After its assembly with the viral DNA, integrase cleaves immediately downstream of the invariant nucleotide, typically removing 2 nucleotides from the C3' termini of both strands of full-length linear viral DNA, generating a pre-integration substrate with the 3' processed ends. The second step is the DNA strand transfer. In the nucleus, integrase catalyzes a staggered cleavage in the cellular target DNA. The 3' recessed ends of viral DNA are joined to the 5' overhanging termini of the cleaved cellular DNA. The third step is the gap repair. Cellular repair machinery fills the gap, thereby completing the integration process. All integrated provirus terminate with dinucleotide 5' TG and CA3', whereas different genera yield different size flanking duplications based on the spacing of the staggered chromosomal DNA cut. The lentiviral duplication is 5 base pair. In addition to the linear double-stranded viral DNA that serves as the substrate for integration, two types of circular DNA are formed in the nucleus. First is LTR circle and second LTR circle. Although these circular DNAs are not substrate for integration, they provide useful markers for nuclear import of the viral DNA as their formation requires enzymatic machinery present predominantly in the nucleus. Now, retroviral integrase proteins are composed of three structurally and functionally distinct domains: the N-terminal zinc binding domain, a catalytic core domain, and a C-terminal domain. Problems with low solubility and propensity to aggregate hindered process in delineating the structure of the integrase holoenzymes in its active state. However, the structures of all the three domains independently and in various combinations have been solved by X-ray crystallography. So now that we have covered all the molecular and structural details of HIV and its replication and integration process, now let's quickly go through how HIV can be transmitted. So there are a number of ways through which HIV can be transmitted. The first way is sexual contact. Second is pregnancy, childbirth, and breastfeeding from HIV positive mother. Third is injection or drug use using contaminated needles. Fourth is occupational exposure, as is the risk with all the diseases. And fifth is blood transfusion and organ transplant, but that is rare. Now, the basic characteristics of HIV disease course. So, HIV infects and destroys the cells of the immune system, which are the CD4 plus cells. And this virus can infect humans only. And it attacks your CD4 plus cells and then uses them to make copies of itself, as we have discussed in detail. And from the onset of initial infection, it takes approximately 8 to 10 years for the disease to progress and to take over.